That might be a good time where you can take your measurements for curly bottom.
Is that what we're writing our measurements?
In this, I was about to say, in this scenario, do we need separate papers?
Well, just yes, you do, because you each need to enter it into your lab notebook, so.
That's this. I didn't know we were getting hand out for it, so I just started doing it now. It's just. Yeah.
Okay, so Kirby Bauer, what we're doing is we want to measure the zone of inhibition. If you remember, you prepared 4 lawn plates of 4 different species of bacteria. Note that the gram reaction of each one is listed on the top here. So we have 2 gram-negative species, E. coli and Pseudomonas aeruginosa, and then 2 gram-positive species, Enterococcus faecalis and Staphylococcus aureus. And that's important when you start to try to determine if these antibiotics are broad or narrow spectrum. Okay. Now, what you want to do is use your ruler, and we're measuring in millimeters. We want to measure the zone of inhibition, this area of no growth around the antibiotic disc. Sometimes the growth goes right up to the disc, sometimes even over the disc. In that case, clearly that antibiotic has no effect. So we can say that this bacteria, this is E. coli, we could say E. coli is resistant to antibiotic A. Right? That antibiotic has no effect on preventing its growth. It's resistant. However, if we have this zone of inhibition where it clearly has some effect, well, we want to measure it because the size of it determines whether or not that bacteria is actually resistant or sensitive. So you are going to use your ruler and measure the diameter of that zone of inhibition. Now note that this is in centimeters. So if you were to measure, say, 2 centimeters, that's 20 millimeters. So you multiply the number here by 10 to record millimeters. If your disc ended up, sometimes they end up like over here. Right? And so when you have this zone, the circle is kind of cut off, or maybe even the zones of inhibition are colliding with the one next to it, and it's really hard to find this distance. You have 2 options. One, think of, it can be any, like at any angle, right? So in this case, I would not try to measure in this way, but I would try to measure the diameter this way. Or if you could at least get the radius, which is from the center of the disc outward, then multiply that number by 2. Okay? So that's what you will be writing in for zone size under each species, and you're listing the 4 antibiotics on the left-hand side. Once you have that information, you're then going to look at this card. So if you determined for penicillin, let's say, that your zone of inhibition was 15 millimeters, well, you look under penicillin and you see, ooh, greater than or equal to 15 means that that microbe is moderately susceptible to penicillin. If your zone of inhibition was 14, that means it's resistant. Even though there's a zone of inhibition, we have to look at this card to see whether that species is resistant or susceptible. That make sense? So that's what's going in the column next to zone size. When you have all of that information on the right-hand side, you're going to determine is it broad or narrow. So if you find that every species is sensitive to it, all four, then it's broad, right? It's affecting a whole wide range of bacteria. If you find it's only effective against gram-negative species, but not gram-positive, or vice versa, then that's narrow. Now, just for it to be narrow doesn't mean that it has to affect all gram-positive and no gram-negative. So if you find that it's effective, meaning E. coli is sensitive to an antibiotic, where everything else is resistant, it's still narrow. It's still only affecting a small subset of the bacteria. Okay. And then the mode of action of the antibiotic is what was given last class. I listed different ways antibiotics can prevent the growth of bacteria, whether it's damaging the DNA, blocking protein synthesis, and then I gave examples of different antibiotics that work in that way. So look it up. If you have here penicillin, look up how penicillin works. List that there. Is that clear? Okay. So, although I love to kind of like present on something, get you going, and wait until we transition to the next one, this one is the more finicky one with the time restraints, so I do want to unfortunately kind of put that aside and get started on transformation. Okay, so transformation, as we know, is one of the three methods of what?
Gene transfer.
Horizontal gene transfer. And we know that horizontal gene transfer and mutations are the two ways that bacteria establish genetic diversity. Okay. We already did conjugation. We're not doing transduction. This is transformation, the second method of horizontal gene transfer. And this one requires the uptake. This method is the uptake of naked DNA. Now, the method that we are utilizing to carry out transformation is called heat shock. So some bacteria naturally carry out transformation in their life cycle, but we want to force it. We want to induce it in the lab for the purpose of trying to push a foreign DNA molecule into the cell. So I've given examples multiple times of how we use other cells to express human insulin, as well as many different biotherapeutics. So how do you get that gene into the cell? Transformation. Okay. So we start with E. coli. That E. coli is put on very cold calcium chloride, makes it really cold. We add the plasmid. Now this plasmid is foreign to it, right? In order to have this uptake of naked DNA, and by naked we mean there's nothing around it. There's no vesicle around it. There's no capsid like a virus. It's just a DNA molecule. Okay, so we add the plasmid, and then this is our method of transformation, heat shock. We already have it at an ice cold temperature, and we are going to shock it by quickly putting it at very warm temperature. Not so hot that it's going to kill it, but warm enough that it shocks it so that these kind of micro tears form in the cell wall and plasma membrane. Okay. Those openings allow the plasmid to enter. I should mention, the other thing that the calcium chloride does, besides just providing this cold temperature, is what is the charge of the membrane of bacteria? So remember, all bacteria have what charge? 50-50 chance, positive or negative. Negative, has a negative charge. What charge does DNA carry? Also negative. DNA has a negative charge. And so, is it opposite to attract, or is it similar charge to attract? Opposite to attract. So when you have a negative charge and a negative charge, they don't want to go anywhere near each other. Okay? Calcium chloride, what happens is that this breaks up into its individual ions. The chloride ion isn't really used. The calcium ion is positively charged. It coats all of the DNA and all of the surface of the membrane. So it neutralizes it. If two things are neutral, they can mix just fine. Okay? So the use of calcium chloride, we're using it to make it really cold, but also we're utilizing those positively charged calcium ions to coat the negatively charged DNA, the negatively charged membrane, so that they will get in close proximity to each other. Okay, so we heat shock the cells, the plasmid can enter at that point, and then we allow the cells to recover, because this was traumatic experience, right? Like suddenly be exposed to that warm temperature and have those micro tears. So then we have to let it at room temperature for a little bit to let it recover, and then we put it on agar to grow. Now, in order for us to see whether or not this is successful, the plasmid we're using is called P-Blue. So usually when you have a name for a plasmid, you have a lowercase p for plasmid, and then some kind of representation of what that plasmid is offering. This one is offering the ability to produce a blue compound, so that your colonies are actually blue. Okay. Now, in this plasmid, there are two key genes. One is an ampicillin resistance gene. So any bacteria that takes up this plasmid should be resistant to ampicillin. So if you plate it on an LB amp plate, it should grow, right? That means if we plate it on an LB amp plate, any cells that are not transformed should not grow, because they don't have that gene. There's also a LAC-Z gene. It's part of the LAC operon. LAC-Z is the gene that codes for beta-galactosidase. This is the enzyme that breaks down lactose. So if a cell is in the presence of lactose, it can consume it and break it down. Well, the reason we care about that is that scientists have figured out that if we introduce a compound that's similar to lactose, so if you look at the chemical structure, it's very similar but not exact, called XGAL, this is in the media. If beta-galactose is around, so this stands for beta-galactosidase, so if the microbe takes this plasmid in, and expresses LAC-Z to produce beta-galactosidase, it will break down the XGAL in the agar into a blue compound, and the colonies will be blue. So now we have two ways of checking whether transformation occurred. The first one is that they can grow in the presence of ampicillin because of this gene. The second one is that they're blue because of this gene producing beta-galactosidase. Okay. So in the end, we will have five different plates. We have our positive control here, or I shouldn't say our positive control, just our control here, just plain LB, making sure that we can have growth of E. coli after transformation, that we didn't, fully kill our bacteria in this process. We want to make sure we have growth. We want to make sure that they can grow in the presence of ampicillin. And then here, LB with ampicillin and XGAL, we also want to make sure that they can express that LAC-Z gene, produce blue colonies. So we are going to carry out transformation with two tubes. The one we're really hoping will be successful, E. coli with that P-Blue plasmid, carrying out heat shock, hoping it takes it up. But then we're also going to have a negative control, E. coli that we don't give the plasmid to. Because of course we want to make sure that this will give us a negative result, right? That with this negative sample, we don't also get blue colors. Okay. Any questions so far? Okay, so you have on page 3 or so, the protocol. You all have that tray, hopefully, from group. You are going to take one sterile tube, and it should be those, we call them flip cap tubes, that are clear in color, in your little rack. They're about 1.5 mils. Yeah, Shaylin, do you mind holding that up? Take one of those and label it with a plus, and the other with a minus. You can use the red wax crayon to be effective. If that doesn't work,
Yes.
lend you my Sharpie.
If the design around it
Try to also put
intertwines,
something
like, it
else
looks like an
on the
E, see?
tube that you would
gets not
know
like
it's
closed
yours.
off. What does that
Whether
mean? You
it's
still
coloring
measure
part of
design,
it,
right?
your initials,
Yep.
because
Initials.
eventually with
But,
the
like-
heat shock, we're going to have different samples in the warm water bath here.
What do I mark it with this?
Yes.
Yep. Just wanted to make sure it was.
Oh, the wax pencil works.
Say it again?
If it's resistant, it's just the same size as mine.
And then where do you want us,
No.
This is 0,
Yeah,
right?
yeah.
Yep.
and where do you want us to mark it, anywhere else?
Anywhere else? So just so you know, you will have time to do this in between this, like
Sure.
10 minutes, wait 15. I just don't want you to be completely lost on this. And it's up to you.
Well, it's up to both of you, Claire and Yaz, whether you wanna just carry it out on your own or work together.
I don't mind, cause I will still have to give Mari the results from whatever we got, so,
Okay,
yeah, I'm okay with that.
how about you? You want help? Okay.
She said like.
I could be your stand-in lab partner, babe, if you need it.
I know, but it's okay.
Okay, so you mark one plus, mark the other.
She's
Might
telling,
just have
she's reading
some
aloud, so
kind
it's
of
not like you can hear.
drawing or what have you on the tubes to mark it as yours.
Right, we'll mark
Then
it.
open one of the transfer pipettes to get familiar with the volume. You have a little drawing on the side of the book, right? So we want 250 microliters of ice cold calcium chloride. I think it's in the fridge or the freezer. I gotta go get it for you. But 250. Note that the line kind of in the middle here says 0.5. 250 microliters is also 0.25 mils, which is the line right below it. Okay? So when you aspirate to measure it, you are going to bring it up to that line.